Okay, let's get started with the second lecture today. Pleased to introduce Maria Orive, who is also going to be here for all of this week. And she's done a lot of work on life history, evolution, sexual reproduction, and so on, mostly theoretical work. And but she says she's a biologist and not a mathematician. And she's taken over 36 hours to get here from Kansas, which is where she works. 24 or more in the air, right? Okay, good. So welcome, Maria. And yeah.
Okay, so is the mic on? Okay, can you hear me? I actually have a very loud voice, I'm told all the time by my students. So I wanted to say that I'm not a mathematician, and I will probably drive the real mathematicians crazy because I will not be formal, and I will probably use terms that are loosey-goosey, as we say in the United States. So hopefully you will be able to follow along. So today, I'm going to I have no chalk? Where's my chalk? Today I'm going to start with talking about age-structured populations. So maybe I'll write that. This also lets me try my writing. Oh, not so good. Age-structured population. But my advisor always used to say, before you start with anything complicated, start with something simple. So let's start first with unstructured populations and make a contrast. So we're going to start with the idea of an unstructured population. And what I mean by that is just initially, I'm going to pretend that all the individuals in the population are the same. They're all the same. And we're just going to follow the numbers of the individuals, the demography. And then we're going to make a link between the demography, population genetics, and sort of thinking about evolution. So some of what I'm going to say is going to repeat some of the things that you've already heard, but hopefully will still be helpful. So we're going to start with thinking about a slightly artificial population. Imagine, actually, you don't have to imagine, there's a picture here. These are the PDFs that I posted. And you're not going to need to read anything, but there's a few graphs. So this is a figure from a textbook, and in it, it follows experimental data in yeast. And what's being followed is the number of cells in some quantity, some volume of culture medium. And the actual data is given by these x's. X, 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 X. All right. I'm sorry, no. The actual data is given by the dots. I was wrong. The dots. All right. And so we might ask the question, how can we model the number of yeast cells over time? And so there's a few different options. And you've already talked about some of these options before. But let's start with the idea of exponential growth. And we're going to talk about discrete time and also continuous time. So what we're going to say initially is that some constant fraction of the population reproduces each time unit. So in discrete time, maybe I should spell it correctly. Maybe not. Okay. In discrete time, we might think to ourselves, well, at time t, what is the number of individuals in this population? We can write it like this. We have however many individuals were there one discrete time unit ago, plus some new number of individuals, right? So new individuals. And what we can say is, let's say some constant fraction multiplied by the number of individuals that were there before. Okay? So that's just an intrinsic rate of increase, so we can rewrite that. As this recursion equation. We can also write the difference equation. Something like that. What am I ignoring? real process that happens to real organisms am I completely ignoring? Death, yes. This is a magical yeast culture where no one's dying, so there's no death. I'm ignoring death, and this is exponential growth, so my population size is going to get bigger and bigger and bigger. So if I go ahead and say, well, n at time 0, oh sorry, so loud, is 10, and r is 0.7083, and I graph that, now I get these x's. So the dots are the real data, and the x's are at time 1, here's time 0, we're at 10, time 1, time 2, time 3, times 4, 5, 6, so like that. So you can see that at the beginning, the x's follow the real population, the experimental population from a real laboratory, really well, but later, terrible, not good at all. The fit becomes bad. And that reminds us always that whenever we do models, over some range of parameters, things are probably likely to be true, and over other ranges, they're likely to be not very good. Now, we already talked a little bit about the importance of discrete time versus continuous time. So I like discrete time myself, because I don't know why I like it, but I just do. But in continuous time, we can do the same sort of thing. You might think to yourself, continuous time might make more sense for yeast, which are maybe dividing continuously. The data itself was censused discretely. So the experiment took samples from the experimental population at particular time points. So the data is discrete, but we might imagine that the yeast themselves are not discrete, that they're having overlapping, they're continuous. But nevertheless, we could do something similar. I've put a subscript in a very sloppy way to indicate that this is a different rate of increase here. This is the intrinsic rate of increase. On a very small time scale. So if I rewrite this like this, and then prove to you that I'm not really a mathematician by saying, I'm going to let n at t, the difference between n at t and n at 10t minus 1, be delta n delta t. And I'm going to say, that's approximately n of t dt. What I did is I sort of just jumped between discrete time to continuous time by taking a very small little time steps. That leads me to a differential equation. Right? Because for very small time steps, if my time steps are very, very, very small, infinitesimally small, then the difference in population size from one time step to the other is very small, and I can write it like that. All right. And then, of course, we can solve this differential equation and eventually get some math happens. So here's one equation. And here's another. If I take many time steps in this discrete case, I can eventually write it like this, which I forgot to do earlier. So that I'm writing the population size at some time in the future as a function of the population size at the very beginning. That's the same thing I've got here. And this naturally leads to the fact that the relationship between these two growth rates, when we move from the continuous to the discrete case. All right, well, that's another case. So if I use this equation, and again I say I start with 10 cells, and now I have a slightly different growth rate. graph that, that gives me this dashed line. Right? So the discrete case are the x's, and the dashed line is the continuous case, and both of them fit quite well at the beginning and terribly later on. So none of you would be surprised if I said, well, I need a better model, right? I need something else. What do I need to take into account what's happening to my yeast? If I look at the dots. I'm sorry? Well, they're not dying, because no one's dying. No one ever dies here. Stagnant, yes. It's sort of the reproduction, whatever growth rate they're at, it starts off very fast, but then it goes away, right? And we, biologically that makes sense to us. It might be that the growth media is getting used up, or it's crowded, who knows what's happening. So oftentimes, instead of exponential growth, although we'll mostly think about exponential growth, we think of something else. We think of logistic growth. And we can introduce another parameter, something called a carrying capacity, which defines how quickly this process sort of saturates. And again, in discrete time, we'll write it this way. The number of individuals at time t is, again, the number of individuals that were there before. No one's dying. And we have a term. Well, I'm going over the line, so I think I need to. Let's try that again. Okay. The number at time t is those that were there before. Plus we have the same sort of growth we had before, but now we're going to introduce a term here that looks something like this. There's different ways to do it, but this is a very standard way, where k is what's sometimes called the carrying capacity. Right? And this lets us have a slightly different shape. We always say, oh, well, what are the extremes of this? We can imagine when n is small compared to k. When n is very, very small compared to k, then k minus nt minus 1 over k is close to 1. And then you get exponential growth, which is what we want. But when n is close to k, term goes to 0, and you get no growth. So that is exactly what we wanted. Because we see when n is small at the beginning, it matches exponential growth very, very well. But when n is large, and you hit whatever the carrying capacity is, the growth is, there is no growth, and the population size is just going to some constant value, right? So we can also do a continuous time version of that. Solve that. Do I really want to solve it? Nah. The continuous time version for this, and the handout that's posted goes through the derivation, if anyone's interested. Where C is a constant defined by the initial conditions, right? So if we go ahead and let the parameters be the same as before, but do a carrying capacity of 665, in this case, and do the continuous version, this line is this function. And you see that it fits the yeast perfectly. It's beautiful. Which, of course, we picked the yeast because they were going to behave nicely. So these are very simple models of how populations might grow. And you guys might imagine a whole bunch of other possibilities, right? These are just two of the many different population growth possibilities. So this is a very simple demography. So the question is, how does demography influence population genetics? And for that, we need to think about fitness. And luckily, we've already talked about fitness, so this makes my life much easier. So now we are going to, following what we just did before, think of two types of yeast. And because my yeast in this example are going to be haploid, I don't have to worry about diploidy. So we'll just think of them as two genotypes. We'll call them A and B. And to keep things also simple, we're just going to focus on exponential growth. And we imagine that time isn't going so far that we don't have to worry about the logistic equation. So now, let's think about selection. And try to make a link between the demography and what's going on here, the idea of fitness. So we're going to let, before I was using n of t to be the number of individuals at time t. Now I'm going to keep track, again, kind of sloppily. This is the number of type A at time t. And this is the number of type B. And I'm going to use little a for the growth rate for whatever the A type is, and little b for B. And I could write two equations just like before. And these are the same exponential equations I used earlier. And now I'm just saying, well, I'm just keeping track of the numbers of these two things separately. So there's an intrinsic relationship between demography, where we follow the numbers of organisms, and fitness, because fitness tells us the relative numbers of these different types, right? We'd like to know sort of which one is doing better, which one has higher numbers. So one thing we can do is just say, well, what's the relative frequency of these two types? So I could say, well, the frequency of A is the number of A's at time t over the size of the whole population at time t, which we'll call P at time t. And similarly, we can say, what's the frequency of B? And again, have that just be the relative number. And obviously, these two things have to sum to 1. And instead of following the numbers of individuals, we can follow the frequencies. So I can say, well, how does the ratio of their frequency change over time? You can see, just from the definitions here, that this is equivalent to A of t, B of t, which lets me write it like this. Well, I can just do P0 over Q0. Or now I've just introduced a fitness measure in this discrete time. That's the ratio of these two relative growth rates. So that gives me a measure of fitness. It's actually defined in this way. It's a measure of the fitness of A relative to B. You can take logarithms of both sides, write it in maybe a more standard way. And when you write it this way, you can see that if I plot the log of the ratio of the two frequencies against time, the slope of that line should be the fitness. And so I think I've got off the end. So this figure shows two different strains. And later on, it shows the slope of those two things. So when we define fitness in this particular way, for this very simple haploid model, the fitness W tells us something that we want to know. It tells us how the two types will change in frequency over time. And in this particular case, if W is greater than 1, then A will increase in frequency. If W is less than 1, A decreases in frequency. And W equals 1, they shall have equal fitness, and there shouldn't be a change in their relative frequency. So this particular definition of fitness, and we talked about lots of different definitions of fitness, has this relationship. The continuous time version of this ends up being quite similar. And you can go between the two this way. And in this case, if M is greater than 0, A will increase. If M is less than 0, A decreases. And if M equals 0, there's no change. And if we graphed, again, if we graph the logs of these, we could use the slope to be a fitness. So that immediately tells us something about how selection might act. Because what we can do then is ask the question, well, given this this demography of these two populations, how will the frequency of an allele change from one time unit to the next under this kind of selection? And haploid selection is very easy, which is nice. So in population genetics, we often want to know what's the change in allele frequency. So let's ask the question, what's the change in the frequency of, say, allele A? I keep putting my chalk down and forgetting where I put it. So let's imagine discrete time again. In generation T minus 1, and we have two haploid genotypes. Call them A and B. Their frequencies before selection, we'll call them P of T minus 1 and Q of T minus 1. And their relative fitnesses will define the way we just did. The relative fitness of A to B is W, and the relative fitness of B to itself is 1. Right? We're using B as our reference in this case. So after selection, the frequencies are P times W and Q times 1. We need to normalize these for generation T, because we need them to add to 1. So we'll get PT minus 1 W over the sum of the two. And the same thing over here. And then we can ask our question, what happens in one generation you can do some algebra and you end up with something that looks like this. And this is a sort of a typical population genetics result, that under haploid selection, the change in allele frequency is just going to be this quantity. And you can see that W, once again, tells us what's happening. If W is greater than 1, then the change in frequency of A is positive, so it increases. If W is less than 1, the change is negative, so it decreases. And when W equals 1, selection should have no effect. This ignores drift, it ignores mutation, it's ignoring everything, just selection, right? That's all we have. And you can work out the continuous time version as well. So it's very simple to see in this unstructured case where all organisms are exactly the same, except for the genotype that we're keeping track of, what happens after a generation of selection. So what I'm going to do now is ask the question, how can we build a model similar to this in a population where we have some kind of structure? We have organisms that not only differ maybe in genotype, but also in some other aspect of their life history. And an aspect that a lot of us are interested in is age structure. The handout has some problems that you can work through. It has one for this. It's very simple, so I won't go through it. But now let's imagine that we have another population, and we want to keep track of not just the genotypes, the allele frequencies, but we want to keep track of some other aspect. And in this case, the age of the population, the age of the individuals in the population. To be fair to everybody, I'll start over here once in a while too. So there's lots of different ways to visualize an age-structured population, but a really common way is to use something called a life cycle graph. So how many of you guys have used life cycle graphs before? A few? One or two? So one way is to keep track of the possible types of ages and how you move between them. And for this example, I'm going to use something from a textbook by Sally Otto and Troy Day, where they follow a population of stickleback fish. They just follow the females, so we're not going to worry about males and females. We're just going to look at the females. And they assume that stickleback fish have 4 age groups, 4 years. Year 1, year 2, year 3, year 4. They're all dead after 4 years, assuming. And they're censused sort of at the end of the year after reproduction. That's going to be important. So we can visualize that stickleback example with a life cycle graph that looks like this. That is too high for what I want to draw. So the circles indicate what sometimes are called nodes, so those are the different age classes. And then we have arcs or arrows showing how we can move between them. What I've drawn is indicating there's 2 kinds of movement. There's the probability that you survive from one age to the next. But there's also some fecundity, some reproduction. We'll call, the textbook uses M, so I'll use it, M's also. So we have summarized the life cycle of these fish with this diagram, and then we can use the diagram to help us draw recursion equations for the number of individuals now in each age class, because now we want to keep those separate. Yes? At the age, it's year 1. Yes, I'm using this example because Sally used it. So year 1, you census everybody at the end of the year, they've reproduced. So it's after reproduction, so after the first year. So yes, it's a good point because oftentimes we don't have this. Because year 1, and I'll give you an example later, where number 1 will be newborns and they can't reproduce. But for this example, they do. So there's a lot of variation in these kinds of life cycle graphs. But I wanted to use this one because I have all the numbers for this one. I thought it was nice. We can say, well, how do we know? Again, this is sort of like at the beginning of my really simple example where I'm not keeping track of genetic types. They're all the same, but they're different years. So I just want to keep track of the numbers. So the number of stage 1 or age 1 individuals at time t plus 1. Well, how do we get stage 1 individuals. They have to be made by reproduction from the other stages. And this case is very simple. It's the reproductive number for stage 1 times how many we had in the previous time unit. And the same for each age class. Right? So that's all the different ways you can get into age 1. And then the other ages are much easier. The only way you can get into age 2 is by surviving from the previous year. So it's just the probability that you survive times the number of individuals that were there in the previous stage 1. The number of individuals in year 3 is just the probability of surviving times the number of individuals that were there in the previous year in year 2, and so on for year 4. So this is a very, like I said, a very simple example, and we often then gather this into a matrix where we write the vector t plus 1 given by this quantity, where A is a transition matrix. For this particular case, it's a very nice simple one. That's the transition matrix. Now, this is what's called a less matrix. It's also sometimes called a projection matrix, because it lets you take a vector of the number of individuals in the previous time unit, and 1, and 2, and 3, and 4, and project what the numbers will be one time unit later. That's nice. And Leslie matrices only have 2 kinds of non-zero entries. The fecundities that are always in the top, and sometimes this is 0, and then these survivor probabilities that are in the subdiagonal. The M's and the PI's are sometimes called life history parameters. And there are many different flavors of these kinds of models. I've written them so that these life history parameters are constant. They don't have to be constant. And I've also written them so that they are not stochastic. Right? So we have the very simplest flavor here, and I'm going to stick to that. But as you go on and do more work with sort of this type of model, there's a lot of other possibilities that you could do. Right? But this is the simplest case. And for this, it just sort of tells us what happens to numbers. And we can project. The simplest kind of analysis would be projection. So I think the next picture actually will show up. There. So these 2 graphs show, this graph on top shows population size as a function of time for these 4 different age classes, given certain values of this, and I'll give you this in a minute. And then this gives you the proportion of the population that's in each class. Talk about that. So for this particular example, I'll write it here and I'll leave it. So I don't have to keep writing it. That's the particular values that are used in that example. And so basically, if you start with some number of the 4 different age classes, you can use that transition matrix to just do this very simple thing. And that's, again, just demography. It tells you the numbers of things. So one of the things that it lets you do, however, is make some analyses of how things should change over time. And for that, you just use some basic results from matrix theory. So one of the nice things about this particular type of matrix is that the characteristic equation is very, very simple and leads to a really simple result. So for a Leslie matrix, the characteristic polynomial has a special name. It's often called the Euler-Lotka equation. And there's various ways to write it, but in discrete time. Where the L's are the probability of surviving to a certain age, to age class I. So this is P1, P2, through Pi minus 1. And we define L of 1 as being 1. And the lambdas are the n eigenvalues. I mean, you can always find the characteristic polynomial of any matrix, but for this case, it has a really nice, simple form. And that tells us something interesting, because, of course, the eigenvalues of this matrix control how fast the change in the different age classes go. So the leading eigenvalue gives the growth rate for this particular case, which is a really nice result. Now, you may be more familiar with this equation in the continuous form. It's sometimes written this way. I should have a 1, but I'm going to cross the do not cross line. So this is sort of the continuous form of this equation. And here we are starting new boards at time 0. We can always move between them by taking logarithms, right? So that's probably what you guys have seen. If you've taken a population biology class, you may have seen that version of it. But since I'm going to stick to a discrete time, I'm using that. The handout has the derivation of this equation. It's just using the definition of the characteristic polynomial. So there are n. So n is the number of classes. And so there are n roots to this. I think I have that right. Yes. Well, we define, it's because there is no pi minus 1. In this definition, it's, yeah, but it's the probability. P1 is the probability of surviving from 1 to 2. There is no probability here. L1 is set to 1. We just define it as 1. Because it's, yeah. Sorry, yes. Yeah, we set it to 1. It's because we define, you have different versions of this. If we have done what you'd suggested where we don't have this loop, which you can take out this loop in census at the beginning, and then you don't have to define it that way. There's different versions. So the reason that if we're thinking about demography, we like this is because it gives us a sense of the long-term growth rate of the population. It's given by the leading eigenvalue, the leading non-unit. Eigenvalue for A. And much like our definition of fitness before, the leading non-unit eigenvalue tells us if the population increases or decreases. So if we'll call it lambda 1 for the leading eigenvalue. If it's greater than 1, the population is going to increase. Yes? I'm sorry? Yes, it could. It absolutely could. I have some examples where I'm going to show some complex ones. Well, actually, it's the magnitude or the complex conjugate if it's the largest should always. Well, is it possible that the largest is unreal for a Leslie? All the elements are positive, so it shouldn't be. The largest should be real. Yeah. So demographers often just use this. They just say, okay, well, what's the Leslie matrix looks like? What's the leading non-unit eigenvalue? And is the population going to increase or decrease? So that's fine. For this Stickleback example, for this case here, it obviously has 4 eigenvalues. And only one of them is, right, so lambda 1, the one we're interested in. So this population doesn't just grow, it grows quite fast, right? So that's the long-term growth rate. When the population reaches what's called steady state, which we'll talk about in a second, it means that at that point, each age class in the population is going to be increasing by this amount. So this population is growing very, very fast. It's more than doubling. It's a little unrealistic. Nothing is holding back these sticklebacks. So this is just the equivalent, basically, of that very simple model we did for the haploid yeast, but in a population where we're keeping track of more than one type. So we're keeping track of different ages. We can also use other aspects of this matrix derivation to tell us something about what's going on with respect to the different age classes. But why are we doing this? The reason we're going to do this is that later we're going to define a different Leslie matrix for different genetic types. So we assume in the same way that when I went from a simple population that just had one type of yeast and that all were growing at rate R, and we went to a little bit more complicated population where we had 2 types of yeast, A and B, and they were growing at rates A and B, little a, little b. We're going to go from this case where we just have one genetic type, if you like, and they're growing at rate lambda, to ask the question, what happens if we have more than one genetic type? All right, so that's the elaboration that we're trying to work towards. Does that make sense? Maybe? Your roadmap? All right. The 2 other things that people who talk about age-structured demographies often discuss are the stable age distribution and the age-specific reproductive rates. I don't really want to erase that. I'll erase this. So, in the same way that we can decompose the matrix to find the leading, well, all of the eigenvalues, we can find the equivalent left and right eigenvectors. So the right eigenvector and the left eigenvector that correspond to the leading eigenvalue, we sometimes call them the dominant. So I'm going to define this quantity These are the elements of the right eigenvector corresponding to lambda 1. And what it gives is the proportion of the population in age class X at the stable age distribution. Now you can always find the left and right eigenvectors of any matrix But the nice thing about a Leslie matrix, because it has this very simple form, is that you can write the entries of the left and right eigenvector in terms of properties of the growth rate. So again, L of I is this total probability of reaching age I guess for I greater than 1. And because we're letting everybody reach age 1, we're letting that be 1. And what's shown at the bottom graph here is the iteration, using the projection matrix, the iteration of the population, and taking the proportions of each age class. And you see, they start off, there's quite a bit of variation, but eventually, what happens to the population is that each of the 4 age groups ends up being a constant proportion of the population. In this case, I think the solid line is age class, I think it's age class 1, is age class 1. And you can see that age class 1 dominates the population with, just under, I think, 80% of the population being year 1 individuals. And then you see that I think this is age class 2, yeah, and then 3 and 4 are down here. So most of the population are in age class 1 and 2, and a very small amount of the population. Now, this projection was done just by iterating the projection matrix. But you can use this formula to calculate what those proportions should be. I've told you that lambda 1 is 2.75. So you should be able to get the 4 numbers that correspond to the heights of those 4 lines. I might have you do that in a minute. The left eigenvector also is used in the demography of age-structured populations. It's usually, it's defined differently. One way to do it is define it relative to the youngest age class, which is age 1. And we're going to let this is the long-term reproductive value of each age class X. So it tells you in the long term what the contribution of each age class is to the growth or not growth of the population. The continuous version of this is sometimes written this way. And this is a Fisher's reproductive value. So when we think about the demography, at least, of age-structured populations, by just considering the transition matrix, the projection matrix, we can get 3 very important things. We can get the overall growth rate of the population using the leading non-unit eigenvector value, and we can also get, at steady state, sort of in the long term, what proportion of the population is going to be in each age class, and how much of the contribution of new individuals, newborns, is contributed by the different age classes. So you can calculate that again for these 4 cases. And I put that in as an in-class activity. I don't know if I have no idea what time it is. So we're not going to do that. You guys are going to do it on your own, but it's very straightforward. And one thing is you can check your answers for U of X, for the stable age distribution. They should match up roughly with what those lines are. The last thing I want to talk about with respect to age-structured populations, before we go into selection, which will be in the next lecture, is something called sensitivity analysis. And this leads directly to what we think about when we think about selection, because I said, right now, I'm pretending everybody in the population just differs by what age they are. But later, I'm going to want to say they also differ by the entries of their projection matrix. Maybe it could be just a single entry. We can imagine a difference at a single locus that affects the reproductive capability of the year 1 individuals. Or we can imagine a change at a single locus that affects several of these different life history parameters. So we'd like to know something about how changing these life history parameters changes the way the population grows. And then we'll use that again when we talk about selection. So in general, not just for a Leslie matrix, you can do something called sensitivity analysis. And I hate to erase this, but I'm going to. And we can ask, how does lambda respond to a small change in one of the entries of the projection matrix A? I'll call that Aij. It turns out one way to define it is like this. Well, technically it's, well, I'm not going to worry about it. Where the denominator is the scalar product of the left and right eigenvectors. And we've written this, we're just going to assume for right now that the entries of the left eigenvector are real, so, because we're usually looking at the leading eigenvalue, so we're not going to worry about the complex conjugates and all this stuff. We're just going to deal with real numbers, just make our life simple. It turns out that because for the Leslie matrix, this is true no matter what the projection matrix looks like. And in the next lecture I give, I'm going to talk about stage structure populations that don't have Leslie matrices. They have something more complicated. But in the case of a Leslie matrix, there's only 2 kinds of non-zero entries. So there's it's much easier because there's only 2 kinds of sensitivities. So let's ask, what are the sensitivities of the growth rate of the population to the 2 kinds of life history parameters, the fecundities and the survivorship probabilities? So let's focus first on the age-specific fecundities. So in that case, and let's just consider the leading eigenvalue. This thing is very simple then. Lambda. I can rewrite it this way. The denominator, this is the sort of general case, but we can rewrite it this way for a Leslie matrix. And the nice thing is this denominator is a measure of generation time. It's one kind of generation time. What it gives is the mean age of the parents of age class 1 individuals. So it's a very specific type of generation time. Once they're in stable age distribution. So, which makes sense, right? So it kind of comes out of that. The other thing that you can do is consider, well, actually, I'll come back to that. I'm going to just go ahead and write the equivalent one for the survival probabilities. So for the age-specific survival probabilities, again, we ask, how does the leading eigenvalue change with respect to a small change in a survival probability. And this has a very straightforward shape also. I have to do this right. These are J's, sorry. So they look a little complicated, but actually, in this case, you can write them out in a straightforward way. And the nice thing about this is it lets us do a type of analysis that people who study the evolution of life histories have often thought about. So we often think about the idea that it might be that the life history of an organism is such that if you increase one aspect of either survivorship or fecundity, there would maybe necessarily be a decrease in another aspect, so that there are trade-offs within life histories. And so we might ask the question, how do the sensitivities of either age-specific fecundities or age-specific survival probabilities depend as you move through the life history? So this is very important to the evolution of senescence, the evolution of aging, and also the evolution of sort of reproductive investment. So one thing that's often done in this case is ask the question, how does the sensitivity of the growth rate of the population change with time, change with age? So as an example, we might ask, what's the sensitivity of reproduction with age? And we might take the ratio of the sensitivity of the reproductive output of stage I to the reproductive output of stage I plus 1. So is it more sensitive to earlier reproduction or more sensitive to later reproduction? What's more important? And if you play around with those equations, you can show that this quantity for a Leslie matrix looks like this. It has a very simple form. So let's just think about our, and no, I didn't erase it. That's nice. We had a growth rate of 2.75, right? So it's positive. So this population is increasing. So the relative sensitivity of reproduction in stage I to I plus 1 is given by 2.75 divided by these numbers, right? So you can, that's 0.6, 0.3, and 0.1. Yes?
So your sensitivity, delta lambda by delta AJ, does not depend on J, is it?
Does not depend on
J.
J.
Because you have UI, VI?
Yeah, right. So in this case, the I here, maybe I shouldn't have used I, the I corresponds to the age class. So there's just 1, 2, 3, and 4. Yeah, there's no J. Technically, later on, I'll give you another equation where instead of using, we use the specific entries of the AIJs, and there'll be AIJs in there. But the way for the Leslie matrix, we don't have to worry about that. We can just define it. We've defined it so that these are the MIs and these are the PIs, and we don't have to worry about J. But you're right, it doesn't depend on J, the way I've written it.
AIJ has the J, right?
Yes. So that's the general case. So I probably erased it. In general, no, here. This is the general case for any matrix. And next lecture, I'll talk about stage classified matrices. But for the Leslie matrix, we only have 2 kinds of entries. And so we simplify things to just, the only kinds of AIJs that are not equal to 0 are the MIs and the PIs. We've dropped, really, we could call these AIJs. We could do that, right? We could say this is A11, et cetera, right? And I'll do that later. And so this would be A12, right? We could do it, we could use 2 scripts. But because the Leslie matrix has this special form, I dropped the dependency.
Okay.
That's a good point. Thank you.
And your V is the right eigenvector and U is the left one?
Yes.
Okay.
Did I do that right? Thank you. Yes. I don't know. This sensitivity, it is, I don't know that there's a specific name. There's different versions of it. This version, if you look in Caswell's book, I have the first edition, which is maybe 2001. I'll give it to you later. But the Matrix Population Models book by Caswell has a nice discussion of it. That's not the only way that I've seen it written, but that's generally the way that people who work on life history analysis write it.
But for a non-symmetric matrix, the derivatives can be discontinuous, right? I mean, does that
For a non-square matrix?
For a non-symmetric matrix.
For non-symmetric matrix.
Does that show up? I mean, has that shown up in maybe some of your simulations or?
For a population projection matrix, we should have
Smooth dependence on the entries?
Do you mean that it's, I'm sorry, I'm not sure what you mean by smooth dependence. Could you clarify? Right. So I'm trying to think of a case where I would have an age-structured population that would be true for. And I don't know that it would be, but it's possible. So I'd have to think about that. So we can always come up with matrices that don't behave well. I think as long as the entries of the projection matrix are all positive, which they would be for, they're all zero, they're zero. The way that we generally do these projection matrices, we set them so they're positive. So it should be fine, I would think. But we could definitely we could make up an example where things would go badly. These are good questions. Other questions? So what I want you guys to do, because you have so much time, is later on, so this is posted, and I want you guys to go ahead and find these sensitivities. But so the reason we're interested in this, we might think to ourselves, well, what if we could increase survivorship of age class 2 at the cost of some survivorship in age class 1? What do we think would happen? And so later on when we talk about evolution in these populations, I'm just going to tell you the answer. It's going to depend on how lambda is affected, right? Because it's going to turn out that for age-structured populations, lambda is the equivalent of a fitness measure. So we're going to talk about that next time when we talk about age-structured populations. But so these kinds of relationships tell you whether if you had a change that affected a later stage or an earlier stage, how they might be favored. So that's how the evolution of senescence literature uses this. I'm also just going to write one more of these. Well, it's in the handout, so I don't think I'll write it out. Yes? It's contribution 0, yeah. Yeah, so then it just had, well, you could get weird, you'd get some weirdness if you had zeros. But usually, I mean, you could make these sort of arbitrarily complicated, right? Usually people define them so that they make sort of sense, but actually the most common one is exactly what you said, where age class 1 doesn't reproduce, because they're censusing at the beginning rather than at the end. But this example for the sticklebacks, they census at the end, so I was sticking with that. I have plenty of time. So, yes. The first row and the subdiagonal are the only two occupied. No, when I generalize it in the next lecture for stage classified, you'll see that at least for the examples that I'm interested in, the kinds of organisms I'm interested in, they have non-zero diagonals, and also we can get the upper quadrant can be filled as well. We get all kinds of movement. So, and then in that case, the characteristic equation is not the Euler-Lotka, and you can't write simple forms for these, but you can always calculate them numerically. They also change. This is a general for sensitivity analysis, but all the really simple results from the Leslie matrix go away as soon as you have something other than, I mean, there are some versions that are not too complicated, but once you allow it to vary from a simple life history, you start getting too many degrees of freedom and you get all sorts of things. But for a lot of the types of organisms that people traditionally studied, they defined age classes so that you couldn't move backwards, right? You can't move backwards in time. So the key here is that these are time-dependent stages. What I'll do next time is I'll say, I'm going to let them not be time-dependent anymore. They're going to be something else. They're going to be size, or they're going to be developmental stage. And then they don't necessarily depend on time. So the key is that there's only one direction of movement, right? And there's only one kind of reproduction. And it's usually, we imagine it's sexual reproduction. That's what's happening. I actually finished early because I think I went too fast, but that's okay. Are there other questions?
Which you have mentioned in this Leslie matrix, yeah. That is the example you are showing in the matrix. So what's the value, the 2, 3, 4, 4?
That's the number, that's a good, so these are the age-specific fecundities. And the way this was defined is this is censused at the end of the year after reproduction. So it actually includes, you have to reproduce and you have to sort of make it through that year. But that means that for every individual in stage 1, at the end of the year, in the next year, we expect 2 newborns, new stage 1. For every individual in stage 2, we expect 3. For every individual in stages 3 and 4, we expect 4. So that's sort of the maximum fecundity. And this was done in terms of the number of female fish. So this is on average the number of female fish that they took. So this is taken from, from censusing some fish population. Pretending I'm a real biologist here. Like, it's fish. These are just probabilities, right? So it's a probability that given you're in stage 1, at the end of the first year, you'll make it to stage 2. So that has a fairly high probability, but you see it gets increasingly ugly. Just looking at this, if I have the answers, you guys will work them out yourselves. But just looking at, if I asked you, remember the stable age distribution, right? The stable age distribution gave, oh, actually, you know the answer because you can see the graph. It turns out that the stable age distribution for these is maximized for the year 1 individuals at about 0.8, right? And that makes sense because we have all these inputs into that. So that's most of the population at any given time is made up of year 1 individuals. And then year 2, right, we have some survivorship, but then it falls off rather dramatically after that. And that's why these two, the year 3 and year 4 populations are very small. They're a very small proportion of the whole population. Very few individuals survive to year 3 and year 4. Most sticklebacks apparently are just 1 or 2 years old. 1 or 2 years old. Yeah.
Maybe you could say a little bit about the general questions that you mentioned something about size and things like that.
Do you mean for stage structures or for age structure?
So just sort of generally.
So
Yeah.
my
context, but also.
So I'm interested in sort of 2 different things. One is just generally life history evolution, where we do these kinds of, I'm going to show you later a population genetics model where it turns out that the ability of an allele that makes a small change, so we'll do a weak selection kind of model, an allele that makes a small change in some of these entries. And we ask the question, when will that allele increase when rare? And a huge part of that has to do with the sensitivity analysis of the particular life history stage that it changes. So it kind of makes sense in the same way that we talked about the yeast model, right? When would the type A yeast increase? The type A yeast increases when it has a higher fitness. So if we have an allele that causes some changes in these matrices, they'll increase when rare when the life history that they're changing when the growth rate depends more heavily on that life history. So, and that was what I was planning to talk about next time. Yeah, and I should have said that at the beginning. So at the very beginning, we had these yeast, and nobody was dying, and everybody was there. And we talked about censusing a population at the end of the year, at the end of things. But in in many organisms that we're interested in, we have overlapping generations. So if you look at the population at any given time, the individuals that are reproducing are not all the same age, right? There's individuals like these sticklebacks of year 1, 2, 3. Humans are a classic example of an age-structured population, right? You don't have to have the age classes correspond exactly to 1 year. Sometimes sometimes they have the age classes collected. So you can collect more than one age class. And then you have to change these probabilities to sort of take that into account. But a lot of the populations, a lot of the types of organisms that we're interested in have overlapping populations. And so that's when we have age-structured populations.
Maybe just like a comment about yeast.
Yes.
So yeast actually has a lot of structure, which is just like
I was making it up.
This. It has, and in fact, the evolution experiments show more and more that the yeast has a lot of memory of what it did earlier, and they also die as well, and how long they spend sort of starting to grow again depends on what they had in the past. So the sort of general effect of the memory of the population of what's past is the same rate.
So,
plays an important role even in the really simple way.
well, even when we talk about a more general version of this, the stage structured populations, the stage could be something like the number. So in the case of yeast, something that might be important is the number of mitotic cycles it's gone through, which is like an age, since it went through a meiosis. So there's ways that you can structure age structure and stage structure into something like a microorganism as well. I mean, I don't know if they keep track of.
Even in a similar way, even just within 1 week, before it divides.
And after it divides.
Yeah. Divided, when born, grows, a whole lot of things that happen. Sort of multiple stages of things. It's already
the only thing that you need to use this kind of model in your organism, whatever your organism that you're interested in, is that you define some kind of time step. And so your time step determines the values of these. And that time step in this particular case was a year because that was what made sense for these sticklebacks that reproduced annually. The time step doesn't have to be a year. It just has to be some sort of constant time step. When we move to stage structures, you still need a constant time step, but you don't have to move from one stage to the next in that time step. You can stay in the same stage.
I also have a question about the faces. So here you have the sort of phenotype.
This is all phenotype.
I know. So the phenotype will be defined by this face, and the sort of fitness is related to the lambda. And you mentioned that if they change one of these elements of the picture, the gene associated with that will better or worse. Is it also true that if you say change two elements, that those are edited, that the faces?
So you can, so all the models that the models I've done for stage structured populations, I have ignored epistasis. So I have assumed additive results, but you don't have to. It just gets more complicated. But the thing is that most of the time, the work I've done on most of these models have been single locus. So yeah. Yeah. Lambda depends on all the elements, right? And so, right. Well, yeah, in this case, there's always, if you define fitness in this way, you naturally have a framework for epistasis, because every single entry of the matrix affects lambda, right? And so if you assume, which is reasonable, that that allelic changes could change more than one, there's no reason to assume that they'd have to be additive. The analysis I'm going to present is an additive one, just because it was easy. I know. I was so sure that was going to take way longer than it did, but that's, maybe they'll be grateful that I was 10 minutes shorter rather than 20 minutes longer. I don't know. They're like, no.